let me know now. Otherwise, I'll go ahead and get started. And as I get started with the chapter, feel free to interrupt at any time and ask questions. Really, when it comes to chapter 7, I assume that for the most part, these should be review. Because you should have seen this material on Biology 1406. So you should review the general concepts of genome, gene, genetics, genomics. So I'll let you read about that. That's just not very complicated. We do need to go over the structure of DNA. Genome is the entire genetic material of a cell. The genetic material is written in DNA form. DNA is a nucleic acid. Nucleic acids are macromolecules, as you learned in chapter 2. And these macromolecules are made by smaller subunits, which in this case are called nucleotides. In the case of DNA, the nucleotide is made by a sugar, a pentose sugar, called deoxyribose, right here. So this is the sugar. Attached to carbon 1 of the sugar, this right here is carbon 1, is a nitrogenous base right here. The nitrogenous base is a ring of carbons with nitrogens. That's what we call a nitrogenous base. Attached to carbon 5 of the sugar is a phosphate group. So this is a nucleotide for DNA. And we know it's a nucleotide for DNA because the sugar is deoxyribose. So you should definitely know the basic structure of a nucleotide, which you should have learned on chapter 2 anyway. The way that these nucleotides are put together to make a DNA molecule is that the phosphate group from one nucleotide makes a covalent bond with carbon number 3 of another nucleotide. And the phosphate group from that nucleotide, and remember that phosphate group is attached to carbon 5, is attached to carbon 3 of the next nucleotide. So essentially we have carbon 5, to which the phosphate group is attached to carbon 3, then carbon 5, to which the phosphate group is attached to carbon 3. So this is what we call the phosphate sugar backbone of the molecule. And notice that as the molecule attaches in this manner, we're going to have, this is a linear molecule, we're going to have carbon 3 of the very first nucleotide in the structure that's going to be unattached because that's the first nucleotide. And then at the other end, the unattached carbon will be number 5. So what we say is that the molecule has an orientation. It's not the same from one end as it is from the other. So this is the 3' to 5' orientation in this particular example. So DNA is a double-stranded molecule. In this particular depiction, the ribbon is the carbon phosphate part of the molecule, which is depicted as a ribbon. And so we have 2 ribbons, so we have 2 linear DNA structures which intertwine with each other to make a double helix. Notice that if on one of these molecules the orientation is 5', meaning the last nucleotide carbon that is unattached is the 5' carbon, and that same molecule at the other end will be a 3'. The other DNA molecule is going to be opposite. It's going to have 3' at one end, and at the opposite end will be 3'. Okay. So the reason why these 2 strands have to be going in opposite directions is because the bases, the nitrogenous bases, which are attached to carbon number 1, have to be facing each other. So let me go back here and remind you what the carbon number is. We start counting the carbons clockwise from the oxygen. So this is number 1. This is number 1. This is number 2. This is number 3. This is number 4. And number 5 is outside the ring. Remember from chapter 2 that when we have a ring, every time 2 lines intersect, there is a carbon there. So even though there's not a C to tell you, oh, there is a carbon, chemists assume every time you have a corner, there's a carbon. Okay, so just remember that. That's from chapter 2. All right, so let me go back, and we were talking about the need for these bases to be facing each other, and that's the reason why the strands go in opposite directions. And let me give you an idea as to why. If this is one of the strands right here, and let's say this strand is, this is really, give me a second, let me see if I can make this work a little better. Okay, now it's really not working. Okay, all right, so if this is the strand right here, and the strand is 3' to 5', and these are the sugars going in this direction. If the second strand was running in the same direction, 5' to 3', right there, this will be 3', this will be 5'. This is why it's doing this. Notice that the sugars would not be facing each other. In order for the sugars to face each other, one of the strands have to be turned upside down, if you will, so that now it will be in an antiparallel or opposite direction. So that's the reason why the strands have to run in different directions, is so that the sugars can face each other. Okay, so now that one is facing in that direction. The other strand is going to have to be turned in the opposite direction for the sugars to actually face. Okay, so hopefully that makes some sense there. All right, so the nitrogenous bases have to face each other, and the nitrogenous bases come in one of two types, purines or pyrimidines. We have two forms of purines. Sorry, we have guanines and we have adenines. We have 3 forms of pyrimidines. We have cytosines, and thymine, and uracil. But uracil is only found in RNA. So in DNA, we're going to have guanines, adenines as purines, cytosines and thymines and as pyrimidines. Purines and pyrimidines can bind to each other. That's why these bases have to be facing each other, so they can form hydrogen bonds with each other. These dotted lines here represent hydrogen bonds, letting you know these are very weak bonds. That's why the line is not a solid line. Typically, a purine and a pyrimidine bind with each other, and G's are always going to bind with C's and A's with T's. So adenines with thymines, guanines with cytosines. So that's how the DNA double helix is going to be put together. We're going to have 2 strands, antiparallel, meaning if one is 3' to 5', the other one is going to be in the opposite direction, 3' to 5', so that the nitrogenous bases can face each other and form weak hydrogen bonds with each other. Always we're going to have a pyrimidine binding with a purine, in this case an adenine binding with a T, because A's always bind with T's. There's another adenine, so there's another T, and that's another adenine, so that's another T. So in this case, it's just A's and T's. This is another depiction of the same, and in this case, what we have is the same thing, just the nitrogenous bases is showing you how they are antiparallel. Antiparallel means that the strands are running in different directions. One is going 3' to 5', the other one will be going 5' to 3', so that the bases can face each other. Notice that A's and T's bind by 2 hydrogen bonds, versus G's and C's, which bind with 3 hydrogen bonds. Another kind characteristic of DNA is that the molecules are complementary. What that means is that G's always bind with C's, A's always bind with T's, so that if you were to have one strand, you could find what the sequence of nitrogenous bases in the other strand is by putting the complementary base. So the complementary base for A is T, the complementary base for T is A, the complementary base for C is G, and for G is C. Therefore, if you have only one strand, you could guess the other strand by putting the complementary base. If you have an A, you're going to put a T. If you have a G, you're going to put a C, T A, A T, C G, G C. Okay, that's what it means to have complementary bases. So these are the characteristics of DNA. A DNA molecule is going to be antiparallel. The molecule is going to have antiparallel strands, and the bases are going to be complementary. Okay. Now, in the case of prokaryotic cells, DNA is found as one single circular molecule, and you should have learned that on chapter 3. So these single-stranded molecule is a double-stranded DNA molecule, but it's a single molecule. We call it one chromosome. Versus, for example, eukaryotic organisms, which have more than one chromosome, and the chromosomes are linear. In the case of humans, we have 23 pairs of chromosomes. And the chromosomes are linear. For example, we have the X chromosome and we have the Y chromosome. So they're aligned. This is the chromosome when it's been replicated. So in the case of bacteria, what we have is one circular chromosome. And this is a humongous molecule, and that's why it looks like a big blob in here, is because it's huge. So it's a coil and supercoiled circle. We also find DNA in smaller pieces, circular pieces, called plasmids. And I will talk more about plasmids in the next chapter. But plasmids are not a they're non-chromosomal DNA. They're not part of the main chromosome. They're non-essential, they're small, they replicate independently, etc. All right, so DNA is going to replicate by binary fission, and when it replicates, we'll end up with two identical pieces of DNA, which each will be given to a different daughter cell. Notice that DNA replicates always at the same place. That place where DNA replicates is called the origin of replication. Okay, so that little dot there where DNA replicates, that's where replication begins. That's called the OR, or origin of replication. Okay. All right, and we'll talk again. I will talk more about plasmids next chapter. Okay, this is a little E. coli molecule in which the DNA has been spilled, and all of these right there is the chromosome, the single circular molecule. And you can see plasmids here, another plasmid, another plasmid, another plasmid. Okay, eukaryotic DNA is going to be found in two places. It's going to be found in the nucleus. The DNA is going to be linear, and there's going to be one or more than one chromosome. The linear DNA of eukaryotes has more than one origin of replication, so that when the cell begins to replicate, the DNA begins to replicate at each of the origins, creating replicating bubbles. Because DNA is a huge molecule, in the case of both eukaryotes and prokaryotes, it's going to be packaged with proteins to be able to fit inside the cell. In the case of eukaryotes, DNA is also found in organelles. Specifically, the mitochondria and chloroplasts of eukaryotes are going to have their own DNA. The DNA of the mitochondria and the chloroplasts is similar to the DNA of prokaryotes. It is circular. And so it resembles prokaryotic DNA, not eukaryotic DNA. Okay. So, we talked about DNA molecules being antiparallel and being complementary. You should know what that means. Let's talk about DNA replication. DNA contains the recipe on how to make every single enzyme and protein that the cell needs. Therefore, this recipe book, or this recipe encyclopedia, I should say, needs to be copied and given to the daughter cells. The copying process is called replication, and so DNA replication, it's an anabolic process and requires energy and requires building blocks. So it requires nucleotides. Okay. In the case of DNA replication, the synthesis of DNA, the nucleotides carry the energy. So remember that ATP was the energy currency of the cell. Well, ATP is a nucleotide. So every one of the nucleotides can act as an energy molecule. So ATP, TTP, GTP, and CTP are both the building block and carry the energy needed for the synthesis of DNA. So you can see here that during DNA replication, the 5' carbon from one nucleotide binds to the 3' carbon to the other via the phosphate group. You have the phosphate sugar backbone. So the first thing that needs to happen when the DNA is replicated is that these bases, the hydrogen bonds that are bringing together the nitrogenous bases, have to be broken. And that's easy because these are hydrogen bonds which are easily broken. So the two strands are separated. And now we have two templates that the cell can use to make a copy, and therefore it can end up with two copies of the DNA. Remember that DNA is complementary. Therefore, all you need is one template to create the other. So if you have one template, you know that here you're going to need a T, and you're going to need a C, and you're going to need an A, and here another A, and here a T, and now you have one copy of the parent DNA that could be given to one daughter cell. On the other hand, the same thing, the other template tells you what to put in the opposite strand. So there will be an A, and here a T, and a T, and a G, and a C, and now you have the other DNA, copy of the DNA to give to the other daughter cell when the cell divides. So DNA replication can proceed very easily because we have two templates which will give rise to two copies of the original DNA molecule. Now, notice that when DNA is replicated, because the parental strands are separated and each serves as template, they serve as the complementary, as the copy for which the complementary strand will be created. When we have the two strands finished, each of the two strands are going to have a parental strand and a new strand, so that each daughter cell will take with it a copy of the parental strand and a newly replicated complementary strand. This form of replication is called semi-conservative, meaning that the new strands have one old and one new strand. Okay. So semi-conservative defines or explains the characteristic of DNA replication, not the characteristics of DNA. DNA is antiparallel and it is complementary. DNA replication is semi-conservative. So don't let that confuse you. Okay, so if you take, for example, a one parental strand in which only one orientation is being given, you should be able to come up with the orientation and the complementary basis of the second strand. So this is 3 prime, this one will be 5 prime. Because this one is 5 prime, that means that the other strand is going to be 3 prime, 5 prime. And then put in the complementary bases, this will be a C, and a C, and a G, and a G, and an A. And there you have the entire molecule. So expect to do something like that in the test. You should be able to come up with complementary strand given one copy. All right. Now the process of DNA replication requires enzymes. The main enzyme that accomplishes DNA replication is an enzyme called DNA polymerase. This is what we call the anabolic enzyme. So this is the anabolic enzyme, because without this enzyme, the copying of DNA could not be possible. All the other enzymes are helpers, but this one is the main enzyme. So the characteristic of DNA polymerase is twofold. First of all, it can only put nucleotides in their place in one direction. It can only proceed in the 5 prime to 3 prime direction. It cannot grab a nucleotide in the 3 prime to 5 prime configuration and begin to move in that configuration. It can only grab and put nucleotides in one configuration, 5 prime to 3 prime. Okay, and I'll show you the significance of that in just a second. The second characteristic is that DNA polymerase cannot start replication, it can only continue it. So if it has a little fragment already made, it can grab a hold of that little fragment and continue from that point on, but it cannot start replication from scratch. Again, we'll see the significance of that. The third characteristic, which is not here, is in another PowerPoint, is that DNA polymerase has what we call proofreading ability. So the characteristic number 3 is that it can proofread. That is a very important characteristic. Proofreading means edit. It can check for mistakes and edit mistakes. And that is a very important characteristic because it's telling us that the enzyme rarely, almost never, makes a mistake. Making a mistake would mean to put the incorrect base, to match the incorrect base to the complementary base. Okay, that's what a mistake is in DNA language. Okay, so it says that DNA polymerase has to add the phosphate of the new nucleotide to carbon 3' of the existing nucleotide. DNA polymerase adds nucleotides to the hydroxyl group of 3' end of existing nucleotide. Okay, so what this is telling us is that if this is the strand that is being replicated, DNA polymerase will take this is a 3' strand. Okay, so the next one will have to be 5'. So the complementary base will have to be 5'. Let me make this as the base. Okay, will have to be a 5' base, and it will have to be a T. So it will have no problem coming up with the nucleotide in this case, because the nucleotide has to be a 5' to nucleotide in order to fit with this one, which is 3'. Okay. So as long as it moves in the 5' to 3' direction, DNA polymerase has absolutely no problem. What the enzyme cannot do is go in the 3' to 5' direction. Okay, let me go ahead and see if I can explain these a little further. This is the black strands are the parental strands, which are going to be replicated. Imagine that this continues on as a circle here. And this is the other molecule right there. So this is a bacterial DNA, which is a circular DNA. And the origin of replication is right here. This is where replication will begin. And replication will proceed from that point on both directions. So we say that DNA replication is bidirectional because it proceeds from both directions from the point of the origin of replication. The origin of replication is the place where replication starts. And from that point, replication proceeds from right and left from both directions. Okay, so from the OR, this is the 3' strand, this is the 5' strand. In order for DNA polymerase to be able to copy the 3' strand, it's going to have to be moving in the direction away from the origin of replication. That would be the 5' nucleotide, and it will proceed 5' to 3'. The last nucleotide put in will be a 3' nucleotide. Okay, so there is no problem there. Over here, this is a 3' strand, so DNA polymerase will have absolutely no problem putting a 5' nucleotide right there and proceeding 5' to 3', 5' to 3'. The last nucleotide that's going to be put in is going to be a 3' nucleotide. Again, no problem with that. Okay. Now, let's take a look at the other strands. The other strands are a different story. This strand is 5'. If DNA polymerase is going to proceed away from the origin of replication, it would have to be moving 3' to 5', because this one is 5 to 3, the complementary will be 3 to 5. Well, DNA polymerase cannot do that. It cannot move 3 to 5. It has to move 5 to 3, which means that when it comes to replicating this strand right here, it's going to have to go in the opposite direction. That way it's moving 5 to 3. Okay. Same thing is going to happen with the opposite strand here, which is again 5 to 3. DNA polymerase will have to be moving towards the origin of replication because it has to move 5 to 3. Okay. So notice that when it comes to replicating DNA, you're going to have two strands being replicated on one side of the origin of replication, two strands being replicated on the other side of the origin of replication. One of the strands will move away from the origin of replication, the other one will move towards the origin of replication. And the same thing will happen on the other side of the origin of replication. One strand will be replicated towards the origin of replication, the other one will be replicated away from the origin of replication. Okay, so also notice that the replication begins with this little blue line right here, indicating that that is not made by DNA polymerase. That little blue line is a little short piece of RNA made by an enzyme called a primase. Remember that one of the characteristics of DNA polymerase is that it cannot start replication from scratch. Cannot start DNA replication from scratch. It can only add to an existing, already replicated piece. So the cell needs to make a little piece of RNA using an enzyme called a primase, which makes a tiny little sequence of RNA right there. And now DNA polymerase can grab a hold of that already made sequence and continue from that point on. The little piece synthesized by the primase is called the primer. So each piece of DNA that's being replicated will start with a primer. Okay, so that kind of complicates things. Notice also that as DNA polymerase replicates, one of the strands is going to be replicated without a problem. So we have the strand that's being replicated right here, 5' to 3', there's the primer. DNA polymerase grabs a hold of the primer and continues uninterrupted until it meets with the other end. So that part is no problem. Same thing on this side. the DNA polymerase that's replicating the 3' to 5' strand can move in the 5' to 3' direction. It can replicate without any problem and continues on until it meets with the other side. Okay. The others, however, have a problem. The DNA polymerase that's replicating 5' to 3' going towards the original replication will grab a hold of the primer and begin replicating. And all of a sudden bumps against the strand on the other side. So it has to detach, go back, make another primer, begin synthesis of the strand again, of the replication, then bumps against the previous strand, detaches, goes back, another primer's made, DNA polymerase grabs a hold of the primer, continues replicating, bumps against the previous strand, and it constantly has to go back and back and back. Okay? So the same thing happens over here with this other strand that is moving towards the original replication. So as DNA is being replicated, one of the strands will be replicated continuously, the other one will be replicated in a lagging or discontinuous manner. So we have a lagging strand, we have a leading strand. Okay. And the reason why we have lagging strands and leading strands is because DNA polymerase can only proceed in one direction. Okay. So again, that's another complication. Also notice that when DNA is replicated, 4 DNA polymerases are going to be needed. 1 for each side, 2 for each side of the each side of the OR. So 1 for the leading strand, 1 for the lagging strand, and then on the other side, another one for the leading strand and another one for the lagging strand. So that's 4 DNA polymerases total that will be needed when DNA is being replicated. So here's an example of what happens at what we call a replication fork. So the origin of replication is not being depicted in this picture, but it would probably be somewhere in here. And the origin of, so there is another fork on the other side, which unfortunately is not being shown in this picture. And it's unfortunate because it makes the picture inaccurate. But this picture is from your book. What the picture is trying to show you, however, are all of the enzymes needed for DNA replication. And that is something you need to know. You need to be able to come up with all the enzymes needed for DNA replication. Again, the main enzyme, the one we cannot do without, is DNA polymerase. That's the anabolic enzyme. That's the first one that should come to mind when we think of DNA replication. So there's DNA polymerase right there. Okay. Now we also need helicases, and helicases, and right there, DNA helicases are enzymes that are going to unwind DNA. So DNA is a double-stranded helix. It needs to be unwound and separated. So the helicases will do that, separates the DNA, untwists it, separates it, and then stabilizing proteins sit on the separated strands so that they are not going to come together again. Now the strands are separated and DNA polymerase can begin to work. However, we also need a primase. So we need helicases, we need stabilizing proteins, we need a primase to make the primer, right here, the primer. Oops, the next one. Okay, there you go, the primer made by the primase. And then to top it all off, after DNA replication, the cell needs to be able to cut the primer away, fill it in, and the little gap with another DNA polymerase with DNA, and then there has to be a little enzyme that glues the little gaps together. That enzyme is called a ligase. So we're going to need ligase that is going to join the fragments. And by the way, the fragments are called Okazaki fragments. We're going to need DNA polymerase. We're going to need RNase H, which removes the primers. We're going to need helicases to unwind the DNA, we're going to need stabilizing proteins to keep the pieces apart. So these are the enzymes and proteins needed. We're missing ligases, and we need ligases too. Okay, so we need DNA polymerase, that's the anabolic enzyme. Okay. We need the ligase to make the primer. We need helicases to unwind the helix, stabilizing proteins to keep the two strands apart. We need RNase H to remove the primers. We need ligases to glue the fragments together. So as we go looking here, this is, I have no idea what that is. We can make it bigger. All right, I don't know why this is so tiny. I cannot read it. It's okay. All right, so that's what I was looking for here. All right, so the little primers need to be removed by the RNase H. The pieces that are being removed now can be filled by DNA polymerase, because DNA polymerase will have something to attach to. The little pieces that are being made are called Okazaki fragments. So this is one Okazaki fragment. Here's another Okazaki fragment. There's another Okazaki fragment. The little Okazaki fragments are glued together by ligases. And again, you should make a list of all the enzymes needed and their function, okay, for DNA replication. And that's another depiction of the same. Here you can see the Okazaki fragments. And in this case, you see two replicating forks, which is typically what we see. Okay. By the time DNA is replicated, after DNA is replicated, something else happens. Bacteria are going to methylate DNA. And methylation means that a CH3 methyl group will be added in strategic places in different parts of the molecule. This is a way of branding the molecule. Each cell will have their own, each species of bacteria will have their own unique way in which they brand their DNA so as to recognize it as their own DNA. And that's important because sometimes bacterial cells are going to be invaded by viral DNA, and the cell must be able to tell what is their DNA and what is a foreign DNA, and methylation will allow them to do that. Viral DNA will not have a methylation pattern like the bacterial DNA will have. So it's a way of protecting themselves. DNA polymerase between eukaryotes and prokaryotes is very similar. There isn't much difference between them. One of the differences, however, is that DNA polymerase in bacteria works much faster, 1,000 nucleotides pairs per second versus 50 nucleotides pairs per second in eukaryotes. Bacterial DNA is much faster. Bacteria have a smaller DNA, so they're going to replicate their DNA within about 40 minutes or so. In the case of eukaryotes, it may take longer, but because there are several origins of replication, ends up taking it about an hour for the whole replication to occur. So even though it's a humongous DNA, because there are many ORs, it proceeds faster than it would otherwise, even though the DNA polymerase is much slower. In the case of bacteria, there's only one OR, but the DNA polymerase is faster, and it is the DNA is not as big. Again, DNA polymerase has proofreading ability, which means that it can correct its mistakes. So if it puts the wrong nucleotide, it can detect that, go back and fix it. So it checks for mistakes. It can, obviously it's not perfect, it can still make mistakes, but it's extremely rare. Mistakes happen 1 in 1 trillion, okay, let's see, that's a million, in 1 billion nucleotides put in place. Okay, so mistakes are extremely rare. Okay, so you need to be aware of that. We call these 1 in 10 to the 9. Okay, 1 in 10 to the 9 nucleotides could DNA polymerase could make a mistake and not catch it. Okay, and that's going to become an important concept in the next chapter. When DNA polymerase makes a mistake and doesn't catch it, that constitutes a mutation. And it is what we call a spontaneous mutation, as we're going to see next chapter. Okay. Questions? Okay, so if there are no questions, I'm going to move on to the next function. And we mentioned that genes, or the chromosome of a cell, is like the library where all of the recipes to make all of the proteins and the enzymes that the cell needs are written. And the recipes themselves is what we call a gene. So that's what we call the functional unit of the cell. And in order to make proteins, the cell is going to go after the recipe and follow the recipe. What the recipe is telling the cell is which amino acid to put, which sequence of amino acids need to be put together to make a specific protein. So the gene is telling the cell the sequence of amino acids needed to make each specific protein. So each gene is different because it codes for a different protein, and each protein has a different sequence of amino acids. So that is one thing. So in order for the cell to be able to make proteins, it has to be able to take the recipe to the organelle within the cell where the protein is made. The organelle in the cell where the protein is made is the ribosome. So ribosomes are the organelles where protein is synthesized. Okay, ribosomes are actually made of RNA and proteins. So take a look at what the problem that the eukaryotic cell has. In the case of the eukaryotic cell, the recipe books are located in the nucleus. The ribosomes, or the kitchen where the proteins are going to be cooked up, are in the cytoplasm. So we have here a problem. The recipe is in one place, the kitchen is in another place. The way the cell solves the problem is by making a copy of the recipe. So the cell is going to copy the recipe. It's actually going to make more than one copy, and then send that copy onto the ribosomes. And the ribosomes can begin to use that copy of the recipe to make the protein. Now, in the case of prokaryotes, we don't have a location problem, but we do have a logistics problem, because even though the recipe books and the kitchens are all in the same compartment, the recipe book is humongous. It's a complete encyclopedia, and it's extremely difficult to try to find the recipe that you want, pull it out of trying to get that little place out and bring it close to the ribosome for it to read it. So again, it's a logistics problem. It's not practical. So the cell is going to do the same thing. It's going to copy the recipe and then send the copy, which is a much smaller piece than the entire DNA, send it to the ribosome. Okay. The process of copying a gene is called transcription. Transcription means to copy. To transcribe something is to copy. To write it again. Transcription. Okay. So transcription is the process that copies a gene. This is a much simpler process than replication, in that there's only one enzyme involved. The enzyme involved is called RNA polymerase. Okay. Transcription can accomplish three things. Transcription can make a copy of a gene, in which case the transcript, the copy of the gene, is going to be called a messenger RNA. And by the way, the copy of the gene is made in RNA language, not DNA language. And again, that makes sense because RNA is cheaper to make and it is less stable. If you're going to make a copy of a recipe, oftentimes you're going to make it with pencil. And any scratch of paper will do, because it's not a permanent copy. It's just a copy that you're going to use temporarily. So it's okay to use RNA. So the cell doesn't invest making a copy that is temporary using DNA, which is precious. Instead, it's going to make just RNA. Okay, so it's going to be an RNA copy, and it's going to be called messenger RNA. Now, RNA is also used to make ribosomes, and the recipe to make the ribosomes will be written in DNA form. So the ribosomes will have to be, in order to be made, the cell has to copy and make an RNA version of the ribosome gene and then fold it into a ribosome. This is what we call rRNA. So transcription is also going to produce ribosomal RNA. And then there's another ribosomal molecule that's going to help in the making of proteins, which is called transfer or tRNA. And again, tRNA will be made and folded into a tRNA. Okay. So transcription makes three things. It makes ribosomal RNA, it makes transfer RNA, and it makes messenger RNA. Out of the three, only messenger RNA is going to be taken on and translated into a protein. Ribosomal RNA will stay as ribosomal RNA. Transfer RNA will stay as transfer RNA. So you should know the three products of transcription, and you should know that only one of them is translated, made into a protein, which is messenger RNA. rRNA and tRNA are not made into proteins. They just stay as ribosomal RNA, this is ribosomal RNA, or transfer RNA. Okay, transcription is pretty straightforward. What we do is we start, we continue, and then we finish. You should know the location though. It happens in the nucleoid of prokaryotes, happens in the nucleus of eukaryotes, and the mitochondria and chloroplasts of eukaryotes. Remember, mitochondria and chloroplasts have their own DNA. The products are messenger RNA, ribosomal RNA, and transfer RNA. You should definitely know that also. The steps: initiation, elongation, termination. The enzyme is RNA polymerase. RNA polymerase does not need a primer. It does only synthesize in the 5' to 3' direction, just like DNA polymerase, and it does not proofread. So if it makes a mistake, the mistake stays, but that's okay because these are temporary copies, so it's okay if there's a mistake here or there. The transcription begins in a place in the gene called the promoter region. So if this is it right here, at the beginning of the gene, there is a sequence called the promoter, which is the place where RNA polymerase binds. RNA polymerase binds to the promoter, and it itself has the ability to unwind the DNA and separate it. So it doesn't need helicases or stabilizing proteins. Once it separates DNA, then it can begin to make the copy of the DNA. Keep in mind that because it can only work in the 5' to 3' direction, it will use the 3' to 5' template piece of DNA to copy. So let me go back here to make that point. All right, so we have the two parental strands. This parental strand is 5' to 3'. The other one is 3' to 5'. Okay, the one that is being copied is this one up here, the one on top, because the cell can only proceed 5' to 3', so it has to copy the 3' to 5' strand. The sigma factor is a little piece that is present in prokaryotic RNA polymerase that allows the cell to bind to the DNA, allows the RNA polymerase to bind to the DNA, and then as soon as it binds to the DNA, the sigma factor is released. Let's see. So you should know RNA polymerase binds to the promoter, moves from there, and begins to transcribe. Transcription, it is a process that happens utilizing many RNA polymerases so that many copies of the messenger RNA can be made. So we have a multiple number of RNA polymerases transcribing the gene at the same time. By the time the RNA polymerase hits the end of the gene, a couple of things can happen that will terminate transcription. One of the things could be a hairpin loop that will pull RNA polymerase away from the DNA and it will end transcription, or there will be a protein that separates RNA polymerase from DNA and end the transcription. So one of these two will happen to terminate transcription. Because prokaryotes have smaller DNA molecules, they need to conserve space. So in order to conserve space, prokaryotic DNA is what we call polycistronic, meaning that genes that are related will use the same promoter, so that when it comes time for transcription, RNA polymerase will sit on the promoter and it will transcribe all 3 genes at the same time, creating a messenger RNA that has the copy of 3 genes, not 1. This happens because these 3 genes are working together. So for example, if these are genes that have to do with lactose catabolism, one of the genes could be the gene for the protein that brings lactose into the cell. Another one can be the gene for the enzyme that breaks lactose into glucose and galactose. The other one could be the gene that turns galactose into glucose. So all 3 will be needed if we want to catabolize. If the cell needs to catabolize lactose, it's going to need all 3 genes. So when it comes time to transcribing, all 3 genes are transcribed at the same time. Okay, so we end up with a polycistronic messenger RNA that has 3 different genes. The cell knows when a gene begins and when it ends because there are nucleotides that tell the cell this is the start, this is the end. AUG always tells the cell the start. This is the beginning of the protein. UAA means stop. AUG says start of another protein. UAG says stop of that protein. That's the end. AUG is start the next protein. UAA is the end of that protein, of the recipe for that protein. Okay, so it tells you where the beginning of the recipe is, where the end of the recipe is for each of the genes. So you should know what polycistronic DNA is, polycistronic messenger RNA is, and where is it found? It's found in prokaryotes. In eukaryotes, on the other hand, there's another process that is very, very unique. It's called post-transcriptional modification. And post-transcriptional modification is a process that modifies the messenger RNA after it has been made. And essentially what happens is that when the messenger RNA is made, there are pieces of the messenger RNA that are trash sequences. And those trash sequences that mean nothing, code for nothing, have to be removed. These are called the introns. So the introns are being removed, and the pieces that are the good part of the recipe are called the exons, and the exons are put together, and now you have the messenger RNA. So the first transcription gives us a copy that has to be modified. That's what's called post-transcriptional modification. This is, again, a space-saving strategy because depending on the protein that the cell needs to make, and sometimes what we're calling here the intron that is being removed could be the recipe for another protein, which means that when that protein needs to be made, the introns will become exons and the exons will become introns. So by modifying which pieces are cut and which pieces are kept, there's almost an infinite number of proteins that could be made with one single transcript. So this is a space-saving strategy again. In this case, what should you know? You should know that post-transcriptional modification happens in eukaryotes only. You should know that introns are removed, exons are kept. Okay. All right, let's see here. It says give the following DNA sequence, choose the appropriate DNA strand to transcribe and give the messenger RNA. Okay, it says the transcription is proceeding from left to right. Okay, so transcription is proceeding from left to right. The promoter region is somewhere in here. on this side, so the promoter region is somewhere in here. Okay, so the question is, which of the two strands will be used to transcribe? Well, DNA polymerase can only proceed in the 5' to 3' direction. If it uses this strand around here, it would have to move 3' to 5', and it cannot do that. Therefore, the strand that is being transcribed has to be the one above, the 3' to 5' strand, because now it can go 5' to 3'. Okay? So the DNA has been transcribed. Once the recipe has been copied, now it needs to be taken to the ribosome to be made. Ribosomes are made of RNA and proteins. The ribosomes of eukaryotes and prokaryotes are slightly different, as you learn in chapter 3. Ribosomes of eukaryotes are a bit bigger. They are 80S ribosomes, and they are found in the rough ER and the cytoplasm. In prokaryotes, the ribosomes are 70S, and they're found in the cytoplasm and attached to the cell membrane. The ribosomes of the mitochondria are 70S ribosomes. They're the same as prokaryotic ribosomes. The process that makes a protein is called translation. So translation is going to take the messenger RNA, which is written in RNA language, and translate the bases into a sequence of amino acids. So because we're translating from one language to another, the process is called translation. The way that ribosomes translate or read the messenger RNA is going to be by reading 3 nucleotides at a time. Each sequence of 3 nucleotides is called a codon. The correlation between a codon and an amino acid is in the genetic code. So the genetic code is this right here. And the genetic code, notice that is written in RNA language. That's why it has U's instead of T's. So what this is telling you is that, for example, if I have a messenger RNA, and the messenger RNA is A, U, yeah, give me just a second. You have a question? Yeah, go ahead. You have a question, Michelle? Michelle, did you have a question? Okay. I thought you had a question. All right, well, let me proceed then. Okay, so if you have a messenger RNA and the messenger RNA has an AUG, that's just great. And then let's say it has a T, I'm sorry, let's say it has an AUC. Oh, this is no good. Okay. The ribosome is going to read the AUG, and that's the start. Okay. AUC is isoleucine, so the ribosome knows to put an isoleucine nucleotide in that place. So let me give you a better example of these. The way the translation is going to proceed is by using ribosomes as the location where translation happens and transfer RNA as the molecule that is going to correlate the codon with the amino acid. So let me show you how that proceeds really quick here. Okay, this is better right here. So this is how translation begins. The messenger RNA is sitting on the ribosome, and here comes the transfer RNA, and the transfer RNA is going to match the AUG codon right here, and on the other side is going to have the correct amino acid according to what we know by the genetic code. So methionine matches with AUG. The next nucleotide is UUU. So the next messenger RNA, the next transfer RNA that comes along is going to have A, that's going to match the UUU, and on the other side it's going to have the amino acid that AAA codes for. So if you go back to the genetic code, AAA codes for, just find AAA, there we go, for lysine. So lysine will be put in the place where AAA is found in the messenger RNA. So there is lysine, oh sorry, what am I saying, UUU, not AAA, that was UUU. So UUU is proline. The next one, okay, go ahead and move it over here. The next one is, I can read it, ACG, and ACG stands for threonine. So the tRNA will match the ACG and have threonine at the other end. So by reading the codons on the messenger RNA and having the tRNAs available with the correct amino acid attached to them, the protein is being made. When it comes to the test, you will be provided with a genetic code like this one, so that you can read the codons and the amino acid that each codon stands for. Okay. So you will be given a sequence of DNA, and you will be asked to transcribe and translate that sequence of DNA. So in this particular example, if this is a DNA, this is 5 prime, this is 5 prime, 3 prime. So when it comes to transcribing, if the promoter is here, which this particular example doesn't give you what the promoter is, if the promoter is on the left, then that means that the piece of DNA that's going to be used for transcription is going to be the one on top right here, because it's the 3 prime to 5 prime. So the messenger RNA is going to be 5 prime, A, U, G, then CCC, which will be G, 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 then GCG, which will be CGC, then TAG, which will be AUC, and then ATT, which will be UAA. So we're going to have AUG, GGG, CGC. Once you have transcribed, then you can use the genetic code to translate. So now you go back to the genetic code to translate the messenger RNA. So AUG was methionine, so you know the first amino acid in the protein is going to be methionine. The next one was GGG. GGG is glycine. So the next amino acid is glycine. The next one was CGC. and CGC is arginine. Okay, and the next one is AUC, which is isoleucine, and the next one was UAA, and UAA is stop, so that's where you stop. Okay, so you should be able to transcribe and translate a piece of DNA. Okay, polyribosomes is again in prokaryotes. More than one ribosome is translating a messenger RNA because we need many copies of a protein, and so we create what are called polyribosomes. Okay. Termination means the end of a translation, and that just happens when the stop codon is reached, and then the ribosomes fall apart, and the tRNA also leaves the messenger RNA. Okay, and this, you should know what transcription happens in prokaryotes versus eukaryotes. Happens in the cytoplasm in the case of prokaryotes. In eukaryotes, happens in the cytoplasm and in the mitochondria. Okay. Last part of the chapter talks about gene expression. And this is a little bit complicated, but actually, let me go ahead and finish it up and go over it since we just have a few more PowerPoints left. Okay, so first of all, not all proteins are needed at all times. The cell only needs some proteins sometimes, other proteins might need them once in a while. So the proteins that are always needed, those genes are always being expressed, are always being transcribed and translated. We call that constitutive genes, the ones that are always being used. The ones that don't need to be transcribed and translated had to be halted. So there has to be some type of control of which genes are transcribed and translated, which ones aren't. The control is always exerted at transcription. In other words, why make a copy of a recipe that we're not going to make? So typically the control happens at the level of transcription. There are three types of enzymes or genes. We have the constitutive enzymes or genes, which are always being made. These are the enzymes that are always needed. Inducible enzymes are enzymes that are always off. These are genes that are not needed, but if we do need them, we can turn them on. Versus repressible enzymes, which are genes or enzymes that are always needed. The genes are always on, and if we need to turn them off, we could turn them off. So inducibles are always off, but we could turn them on. Repressibles are always on, but we could turn them off. In order to understand inducible and repressible genes or enzymes, we need to understand how genes are put together. And the way that genes are put together is with a control region. So we talked about the promoter region as the place where RNA polymerase binds. In front of the promoter region, there is a control region called the operator region. And in front of that will be the actual genes. Okay, and again, these are bacteria, so the genes are polycistronic. There's more than one gene within the same region. So this entire structure is called an operon. So we have inducible operons, which are always off, but they could be turned on. We have repressible operons, which are always on, but it could be turned off. So we have genes that are always off, but they could be turned on. We have genes that are always on, but we could turn them off. So the question was, how does the cell do that? How does the cell turn a gene on, and how does it turn it off? The first operon that was studied was the tryptophan operon and the lac operon. The tryptophan operon is a repressible operon. It's an operon that is always on, but it could be turned off if needed. The operon, the genes, code for enzymes needed to make an amino acid called tryptophan. So the making of tryptophan requires 5 enzymes, and all 5 enzymes are under the control of the same operator and the same promoter. Because this gene is always on, typically there's nothing on the operator and the promoter is free. RNA polymerase binds to it, and these genes are constantly being transcribed and translated. The cell is constantly translating the enzymes to make tryptophan. But what if there is an excess of tryptophan, or maybe the cell encounters an external source of tryptophan and it doesn't need to make its own? Now it needs to turn off the gene. So if there is tryptophan present in excess in the cell, now there is no need to make any more. That excess tryptophan is going to bind to an enzyme, to a repressor, which is normally inactive unless it's bound by excess tryptophan. So when there's excess tryptophan, the tryptophan accumulates and there is a tryptophan available to bind to the repressor. The repressor, when bound by tryptophan, becomes active. It sits on the operator side and prevents RNA polymerase from proceeding and transcribing. And that's how the gene is turned off. In the case of the lac operon is an inducible operon. In this case, instead of an inducer, what is going to happen is that our repressor is going to be turned off. So typically the lac operon or lactose operon refers to the series of genes that bacterial cells need to process lactose. And there's at least 3 genes needed for the processing of lactose. However, the bacterial cell, will not use lactose if glucose is present. As long as glucose is present, the cell doesn't need to use lactose. Lactose requires more energy to break down. Glucose is a straight shot, no need to spend any extra energy. Lactose will just sit there as long as glucose is there. If glucose is removed and lactose is given to the cell, that will be the only scenario when the cell is going to turn on the lac operon. Now it has no choice but to process the lactose. So normally, because glucose is available, there is a repressor sitting in the operator region of the lac operon. So that's the normal condition. If all of a sudden lactose is present and glucose is absent, so there is no glucose, the presence of the lactose is going to bind to the repressor and prevent the repressor from sitting in the operator. Now there is no repressor. RNA polymerase can sit in the promoter and begin transcription. So now the gene is free to be transcribed. But again, for that to happen, there should be no glucose and lactose. So glucose is called the catabolic repressor because it's going to also repress the gene, so that the only scenario where the lac operon will be active will be when there is no glucose and there's lactose present. So for example, if lactose is absent, the lac operon is turned off, even if glucose is present, or if glucose is absent. It doesn't matter. Lactose is gone. Lactose is the one that inactivates the repressor. Therefore, as long as lactose is absent, there will be no messenger RNA transcribed from the lac operon. If lactose is present and glucose are present, again, there's going to be no synthesis, no transcription going on, okay, of the lac operon. If glucose is absent and lactose is absent, again, the lac operon is off. The only time when the lac operon is on is when the glucose is absent, lactose is present. Now we have the lac operon on. And there is transcription of the messenger RNA of the lac operon. And that's basically what this last part of the chapter is about. What you need to know about these, you need to know what we mean by inducer, by a repressor, what is the inducer in the lac operon, what is the repressor, what is the inducer in the lac operon, which is lactose. The repressor in the trip operon is tryptophan itself. And what's a constitutive gene? And in which scenario is the lac operon functioning, which is when lactose is present and glucose is absent. Okay, so that's it for chapter 7, and it's been over an hour. So what I would like to do is schedule